So now we got mutations. Because things could go wrong. Okay? You need to know the names of mutations. And for the most sense, I hate to use the word sense, but we'll be using the word sense for this. But for the most sense, they kind of make sense. Alright? Deletion, translocation, these kinds of things. And there's other ones that actually I'm laughing when we use the word sense because there's missense, nonsense. We'll get into that too. But things could go wrong. Alright? And that can lead to a bad protein, right? It calls for baking soda and you put baking powder in there. It's gonna be horrible, right? So that's what happens over here. Because mom didn't realize that baking soda and baking powder are two different things, right? Alright, so a mutation is a change in the DNA of a cell or a virus. Someone changed something in the DNA. Something got changed in the encyclopedia set. That's why we have different versions of encyclopedia sets. That's why we have different versions of textbooks, because there's some errors in there. I found a few errors in your book, in the questions. I'm sure you have too. That's where we get new additions. Okay? Sometimes it's corrected by certain enzymes. All right? They see it in there. We catch it pretty fast. All right, we bought the encyclopedia set at the library. The library noticed it within a month. It's got a warranty. We can send it right back, and we get the brand new one. Okay? If not corrected, it can be harmful, though. Right? If it says baking powder as a recipe in the main encyclopedia set, but it really meant baking powder, it could be harmful, right? I mean, harmful in the sense of no one wants to eat your bakery or brownies, right? So we can have something called a point mutation. The chemical change in just one base pair of a gene, okay? So it's just one letter is wrong, okay? There's two types. It's either a substitution or an insertion/deletion, okay? The change of a single nucleotide in the DNA template strand can lead to the production of an abnormal protein, like just one letter, okay? For instance, you talked about this, sickle cell anemia, all right? Hemoglobin is what's in your red blood cells, and it's almost 600 amino acids long. It's actually 4 separate chains in one hemoglobin molecule, with about 156 amino acids long. But we're looking at the whole thing, 600 amino acids long, and there's only one difference in it. Now, I don't know why they call it this, but the normal ones are what we call wild type. Okay, the wild type is actually normal. When we get into genetics, you're going to see a lot of that too. We deal with fruit flies and stuff. So the wild type is what we consider normal. And what's happening here is that here's normal hemoglobin, they're calling it wild type hemoglobin, and here's where the mutant hemoglobin is, right? Something, mutation on it. What's happening here in normal, you should have a T here. All right? So the T in the wild type normal is now replaced with an A. All right? So the T is replaced with an A. That means the mRNA is going to read it differently. That one letter. And what will happen is, as you've seen with the genetic code, that sometimes that one letter is going to change the whole amino acids. And in this case, because we just changed that one T into an A, it was supposed to code for a glutamine, but now because it's been changed, it now codes for something called valine. Because of that one simple base pair substitution, the structure of the hemoglobin has changed radically. That's one difference. Okay? These are red blood cells. These are what they're supposed to look like. Nice and round, kind of pale in the center. But with sickle cell, that one base pair difference turns the cells into this sickle banana-shaped thing. And that causes problems because oxygen is going to have problems binding to that. And you can't carry the oxygen from point A to point B, and those people with sickle cell anemia are going to be deprived of certain oxygen in certain areas of their body. So you may have to remove the sickle cells and put in new red blood cells, but that's only going to last for a few months. Okay? So you get those different shaped ones on there. So we have substitution point mutations. Okay? One base, as you just saw, is substituted for another base in the DNA. Okay? The thiamine in that case was substituted for adenine. And because of that one difference, the cells don't look all normal. Okay? Now, what makes someone live with sickle cell anemia, because you're saying, well, it doesn't carry. Well, it's not all the red blood cells. Some red blood cells, I'm just giving you percentages, maybe 60% of red blood cells are normal, but 40% are abnormal. That's a lot. So they have to go through a lot of different situations. We're going to talk about also sickle cell trait. You carry the gene. When you look at the blood, it does. There are some sickle cells with people with sickle cell trait. But it's a very small amount, maybe 5 or 10%. And you can live with 90 to 95% of normal red blood cells and live to a ripe old age of 100. But when it's 60-40, it's difficult to do. Yes? Oh, you're okay? Okay. So we have 3 different subtypes of substitution point mutations. There's something called a silent mutation, where the codon does change, but you've seen from the genetic code that even if we change a certain, we change like the three letter thing on there, some of them will also code for the same ones. Does that make sense? Right? So you could have, let's say, leucine, but there could be 4 different or 6 different codes coding for leucine. So even if there's a base pair change, it might code for the same one. So in that case, yes, you do have a change, but you're still getting the same amino acid. I call up my mother on the cell phone, but I also have her phone number for a house phone. Does that make sense? It's still coding for my mother who's bringing the eggs. Okay? We have also something called a missense mutation. This is where they kind of get weird. A missense mutation. This is where the codon changes and codes for a different amino acid. However, the amino acid does not affect the protein structure much at all. All right? It's maybe a small little wobble, but it's okay. You know, it calls for white eggs. I got brown eggs. It's going to do the same thing, right? Okay? Sometimes this change will do just the opposite effect. It enhances it. Just by using the brown eggs, someone might say, you know what, they taste better. I don't know. They may or may not. Does that make sense? Okay? And then you have a nonsense mutation. My mnemonic is this, is just stop the nonsense. What this does is that the codon will be changed to a certain thing that will cause a stop codon. Those are good. Those are not good at all, rather. Right? They're going to stop it prematurely. It's like someone went to the recipe thing, and after ingredient number 2, it says stop. What about the rest of it? No, just stop. So it's not going to be good at all, right? You have to still put the milk or whatever you want in there. That's called a nonsense mutation. So there's 3 subtypes of doing substitution point mutations. Depends, okay? But you should know what these words mean. So here's some examples of them, okay? This is a silent mutation, all right? The wild type is up here. In other words, this is the normal one up here, and the mRNA is going to be made. The red is the mRNA, and it codes for these amino acids: methionine, lysine, phenylalanine, lysine. Okay? But if there's something here where the G was replaced with an A, then what will happen here is that, yes, you have this here GGU and over here GGC. They both code for glycine. So even though there's a change in there, we're still getting the same amino acid. So you're really not going to see the difference in the protein. That's why it's silent. We don't see it. Does that make sense? Okay. Which I think that's what the evolution of coming up with this, when they found out this genetic code, that's where it evolved better. We just had only 20 different codes for 20 different amino acids. There'll be a lot more mutations that show up. We wouldn't have silent mutations. Does that make sense? But because we have 64, it gives us some leeway, some wobbling movement, justification that we could actually have 6 different codes coding for the same amino acid. Is that clear? Okay? So that's a silent mutation. Here's a missense mutation. Again, the wild type's up here, the normal's up here, but instead of a T at this level, it becomes, instead of a C at this level, it gets mutated to a T, right? To thymine. And in this case, the code here is going to be AGC as opposed to GGC, which means in normal, GGC will code for glycine. In this case, AGC is going to code for serine. That's going to call for a different protein. All right? We call it a missense mutation. And then we have the nonsense mutation. Stop the nonsense, right? And in this case, normally in a wild type up here, normal, you get to stop all the way down here to stop codon. And that codon is still down here. However, you've got a mutation here that a T has been replaced by an A, and now it codes in the mRNA as UAG, which is a stop codon. So you're prematurely stopping this protein from being made. Completed. Okay, does that make sense? You have to play around these. They'll be on your test. Okay. Questions about that? All right. All right, so let's talk about insertion, deletion, and point mutations. Again, these are point mutations. Okay? Insertion, as the name implies, is going to insert or add on one new base pair in the DNA. Okay? The other ones we're just replacing. Does that make sense? We're substituting. This one, we're adding another one in there. This one, or you can have a deletion. You can delete one. Now, this changes things radically. The point mutation that we just saw, where substitution occurs, we're just changing that one over there. And that causes problems, as you've seen. But it only is going to cause a problem with that one amino acid. This causes something we call, it's disastrous. All right? It's worse than what we saw before. Because it actually changes, we call it a frameshift mutation. All right? It alters the DNA by one notch. It moves it over the whole DNA. Let me show you what I mean. Okay? Here's an example. If we have this in a sentence, if this sentence says, the dog saw the cat. Okay? But I want to delete this E over here in that word the. That means the rest of this, if you have to follow this by threes, then everything gets shifted. And look at what it turns into. Now you've got all the amino acids that are going to be forming after that all screwed up. It's not going to be just one. This is why we call it a frame shift. The whole frame is being shifted. And likewise, if I add another letter in there, it gets shifted that way. And it looks like garbage. Does that make sense? A frameshift mutation is much worse, or can be much worse, than the point mutation. Okay? They're all bad. You can see sickle cell, you don't have to do this to get sickle cell anemia. That's bad as in itself. But this is, down the road, it's in the DNA, it's huge, it's disastrous. Okay? So here's an insertion. Okay? Here's where the normal is up here. And we're going to add an extra A in here. That can cause a stop over here. But all these other ones are going to be messed up too. You're adding, you're shifting over that frame. And it could lead to a stop much earlier. We've seen it where a stop, nonsense. You could actually have a stop by just changing a letter, but this is changing the whole DNA by shifting it over. Is that clear? All right. A deletion, we're going to remove. That's normal there. We're going to remove an A, which means, again, it's going to change all the amino acids from here on in. Okay? So it can cause extensive missenses. All right? You could also, now this is an interesting thing. You could have, now we're just talking about one base pair that's just being added in, or one base being removed. But what if you put 3 in there at one time? All right? When you put 3 in there, it may not, it depends on which one it is. It could code for that or not. Does that make sense? If you put 3 in there, if I went back to the other, if I went back to this one over here. All right? We have the dog saw the cat. If I take this E out, it'll turn this. Right? The whole thing will be messed up. And it'll be frameshift mutation. But if I said the dog saw the cat, it'll appear as the dog saw the cat. You can still read it. You just have, because you put a 3 base pair there, the words are still there, but they're moved over. Does that make sense? All right? Play with the letters. The letters, I think, are going to help you. Letters and like words in here and stuff. All right? Other mutations, okay? You could have an inversion. An inversion is 2 or more base pairs, they kind of flip. In other words, here you see the normal, the wild type, you got TG together here, they flip. Now it's GT. That can also cause a coding problem for amino acids. You could have a translocation. I'm going to take, let's say, CAT and move it someplace later on in the DNA. Does that make sense? You do all these, well, you can't do them, you don't want to do them, but this is what happens. Okay? Questions on that? that's what I said, they're not all bad. So, in fact, that's how evolution occurs. Right? The mutation that, you know, the giraffe got a longer neck, it happened with a mutation. Over, you know, centuries for that to happen. But it also could lead to, you know, certain ones couldn't adapt, and those died off. You know, if there's, let's say there's, you know, if certain animals can't live, I don't know, with a lot of carbon dioxide in the air, they're going to die off because they can't adapt. So, yeah, mutations, I mean, you know, we showed the movie X-Men if you wanted to, right? You know, some mutation, you know, I wouldn't do that. But I'm just saying that there's mutations that might benefit, there might be mutations that can cause cancers, right? So, it's a lot of different things that can happen.